Hello dear student, welcome to the memoneet video solution. Now I am Narayana Jat, MBBS third year from the GIMS Gadag Karnataka third year MBBS student. I am here for the SMA of the biology, and today we will discuss about video solution of the PPT 14. So without wasting further time, we will start video solution. So our allotted question today is 1512 200. So read question carefully and give answer. So what is the incorrect for the chromosome number one? So this is question regarding human genome project in HGP. There are chromosome number one. Whenever we do sequencing of the chromosome, first we sequence chromosome number one. No, last we sequence chromosome number one in 2006. So read question. It was the last chromosome to be sequenced, correct? It has a maximum number of genes. Genes are maximum number of genes are present in the one chromosome number one. It was sequenced in 2007. No, it's sequenced in 2006. It is the largest chromosome which is sequenced lastly. Largest chromosome sequence सबसे last में. So question is regarding incorrect. So here statement 2 is incorrect. Now read question 152. The non-human model organism sequence in HGP. Non-human model first sequenced after human are nematode and fruit fly. So nematode and fruit fly is answered directly from here. Now read question 153 carefully. Which enzyme will be produced in the cell in which there is a nonsense mutation in like Y gene? Y gene question regarding which enzyme will be produced in a cell in which there is a nonsense mutation in the chromosome Y. Y gene is having nonsense mutation which does not affect its functional, leading to production of lactose permease. So lactose permease will be produced because here trans-acetylase, lactose permease and trans-acetyl beta galactosidase and trans-acetylase production will be reduced, but lactose permease production will not be reduced. So answer here will be lactose permease because lactose permease and other ZYA. So sequenced by not Y, sequenced by Y sequenced transcriptase and trans-acetylase and beta galactosidase. So here when nonsense mutation occur in the in Y gene, so there is no change in the production of the enzyme lactose permease. So answer is 1. Now read question number 154 carefully. Mark the incorrect with respect to lycopyrrole. Lycopyrrole is discovered by Jacob and Monod. Correct. It discovered by Jacob and Monod is under positive error as well as negative control. Negative and positive both like operon condition occur. Controls catabolic pathway. It's mainly control catabolic pathway. It can also control anabolic pathway also. In this use of feedback, no. Lycopyrrole can't show feedback repression. So now read question number 155 carefully. With respect to the major difference between replication and transcription statements are correct. Transcription is only a segment of DNA. Yeah, segment of DNA. Gene is the segment of DNA which can replicate. Here cystone, segment of the DNA which can transcript are cystone. So here only a segment of DNA will take place. In replication, total DNA is duplicated. In transcription, only one strand of DNA is copied into RNA. That is their statement, the template strand of the DNA. So answer here 4. All are correct. Now read question 156. Replicate splicing mutation. Statement are correct with respect to the splicing. Splicing removal of the intron correct. HN undergo capping and telling. This is capping and to adenyl residue on the third S and known as telling. So here answer is 4. All are correct. Now read question 157. Which of the following process related to reverse transcription? Reverse transcription is formation of the DNA from RNA by enzyme RNA dependent DNA synthesis. So RNA dependent DNA synthesis by RNA dependent DNA polymerase. Now read question 158 carefully. 158. Which of the following structure are present in the core particle of the nucleosome? Whenever we read structure, there is a histone octamer. Around histone octamer, there is the coiling of the DNA, negative DNA, basic histone protein, negative DNA, and non-histone protein also available. So answer is 1. So now read question 159 carefully. So here question 159. Packaging of DNA helix is more complex in eukaryote or prokaryote? More complex in eukaryotes. So it's more complex in eukaryotes than prokaryotes. Directly from answer. Involve polyamines in eukaryotes, polyamine in prokaryotes. Occur with the help of histone protein, non-histone protein. So not only non-histone protein, also histone protein. Required acidic protein that help in the coiling of DNA in prokaryotes. Acidic protein packaging DNA are protein basic nature. So answer is 4 here. Now length of DNA in coli, DNA, length of DNA, number of base pair, here number of base pair 4.6 into 10 raised to 6 and the distance between them. Yani length in when a nucleoside internucleotide distance. So whenever we solve this, you can directly multiply and find answer 1.66 mm. So answer is 2 here. Now read question 161 carefully. Now, in this question, labeling of the protein and isotope labeling of protein and DNA. If you see carefully, labeling of radioactive labeling of the protein is S35 and protein. Now, DNA labeling is protein phosphorus 35, 32, sorry. Sulfur 35 and uh, sulfur 35 and protein, sorry, phosphorus 32, sulfur 35 and phosphorus 32. Now read question number 162. Dominance of RNA world is proved by splicing, capping, polyadenylase, all of the above. So here splicing prove DNA dominance. Only splicing. Removal of the intron. Whenever splicing is mainly process where removal of intron and RNA doesn't have intron. So by dominance, whenever we remove introns from DNA, it's so that we will go to more better side, which is the RNA world, more better product, which is RNA, not DNA. So it's so dominance of the RNA world here in this concept only. Now read 163. Which plant was used to tailor to prove semi-conservative replication at chromosomal level? Whenever Taylor is a scientist which use the one plant. Name of the plant Vishya Phaba. This is the NCRT question. Vishya Phaba is used to for proven of the semi-conservative nature of the semi-conservative nature at the chromosomal layer, chromosomal level. Now read question 164. Unwinding of DNA creates tension released by the enzyme. It's a very well known enzyme, topoisomerase. Topoisomerase enzyme used for their twisting stress, which is the remove twisting stress. So your answer is topoisomerase. Now you should read question 165 carefully. During polymerization of the deoxyribonucleoside triphosphate in bacteria, which of the following enzyme is mainly required? Polymerization of the means formation of the DNA. Which enzyme is required? It's a very common question. DNA dependent DNA polymerase. DNA dependent DNA polymerase. Here for the formation of the DNA means formation of the nucleotide means deoxyribonucleotide triphosphate. Nucleic acid formation here. 166. DNA polymerase catalyze polymerization of or in DNA polymerase. Direction of the DNA polymerase. Very basic question. Direction of the DNA polymerase always 5 days to 3 days. So here the Okazaki segment formation. Here 3 days to 5 days. Here 3 days to 5 days antiparallel structure. So DNA polymerase form directly because 5 days to 3 days a leading strand. But here 5 days to 3 days. Whenever we want 5 days to 3 days, we will go that side. So Okazaki segment formation takes place. So answer is 5 days to 3 days. Now read question 167 carefully. During DNA replication, which of the following does not act as a substrate? DATP act or not? DCTP act or not, DUTP act or not, DGTP act or not. Very easy question. Here, purine and pyrimidine are the nucleotide ways which act as a substrate in the formation of the DNA. So read question DUTP. DUTP, there is DUTP present uracil. U means uracil, and uracil for the formation of the RNA or DNA. RNA. So here we want to formation of the DNA. So this will involve this involve only DUTP not involve. Does not involve. Uracil is here, so so DUTP not involved. DTTP may involve. Now read question number 168. Out of the two strands of DNA, one is carrying genetic information for the transcription and it is called two strand. DNA strand has two. In two strand here, or you can see there are two DNA strand. You know this. Yeah, 5 days to 3 days, 3 days to 5 days, 3 days to 5 days. Anti structure. So which strands directly copy into RNA any formation carrying genetic information in the further inherited type or progeny or DNA to RNA. This is the known as a template strand. So answer is template strand. Answer is 4. Antisense and antisense. This is the template strand or also known as antisense strand. So read question 169. When a mature mRNA was hybridized to its gene, certain loops were observed. These loops present. Any loops formation takes place. Loop is represent introns or exon. It's intron, so exon will be cancelled. And introns present in RNA or DNA, RNA or DNA. Sorry, DNA. So introns in DNA, RNA like intron. So it's so dominance of the and the splicing will take place for the formation of the intron less DNA, and this so dominance of the RNA world. So answer is DNA. Introducing DNA or intervening sequence only. Now read question 170. Poly A is present in HNRNA. You read HNRNA, and there in HNRNA, uh, HNRNA to mRNA formation take place by splicing. There is telling and capping process will go on telling poly A which represent mRNA of eukaryotes because HNRNA mature mRNA will formation in prokaryotes or eukaryotes only to mRNA. HNRNA to mRNA is formation of the polyethyl. This process, splicing all process going on eukaryotes only. So now read question 171. Find out the incorrect match. We should find incorrect match in below. UAA, phenylalanine formation, valine, tryptophan, sense codon, UAG, wow, UVA, UG, UGA. You can read this all are nonsense codon, so this is not sense codon. Now move ahead to question 172. One codon codes for only one amino acid. Hence the code is one codon codes only for one amino acid. This is known as the ambiguous nature of the codons. So ambiguous nature तो present है. So ambiguous or unambiguous. Sorry, when one codon codes for only one amino acid, this is the known as the non-ambiguous. So non-ambiguous, specific and non-ambiguous. So answer is 2. So now read 3. What is incorrect for UTR? So read question UTR. Incorrect about UTR. Provide stability to mRNA. UTR is for the provide stability of mRNA required for efficient translation process, not recognized by any tRNA. So UTR is not recognized or recognized. This will not be recognized. So this will also correct. This one also correct. Present in between the translation unit in mRNA in between or on corner on the end. UTR present on end only. So answer is 1. Question asking about incorrect statement only. In eukaryotes, RNA polymerase third catalyze the synthesis of directly remove RNA in eukaryotes. RNA polymerase third utilize catalyze bypass RNA. tRNA, snRNA. Learn this directly. Now read question 15175. In leg operon, the regulator gene codes for in leg operon regulator gene codes for repressor active or passive repressor. It's a active repressor. So coding by the leg operon regulator gene codes for active repressor. So answer is 4 only. So now read 176. How many locations have been identified in the human genome where single base difference occur? This is the directly NCRT question. 1.4 million locations are found where only one gene difference in the genome project. So your answer is 1.4 million location. So this is the 1.4 million location. So read question 177. Mark the correct one. Forensic science with respect to application, 100% forensic medicine are used all this. Determining the population diversity, determining the genetic diversity. More than one option is correct. Yeah, more than one option correct. Determining the population, we can use genetic diversity. Population diversity may not use this. Nowadays we can using fingerprinting, but not in population. Population demography directly scanning and all other process will go on. Not this process only. Yeah, move ahead. 178. In the, in the technique of DNA fingerprinting, digestion of the DNA is followed by gel electrophoresis. Gel electrophoresis will separate the DNA. After DNA digestion, there is DNA segment formation, and we want to differentiate this DNA segment. And differentiation or can differentiate or differentiation of the DNA segment is take place by the DNA gel electrophoresis. DNA segment will be differentiated. Read the following statement and choose which one is correct. Variation at genetic level arises due to mutation. Correct. Techniques of DNA fingerprinting was initially developed by Alec Jaffrey. Directly NCRT question. NCRT line. Alec Jaffrey first developed gel electrophoresis DNA fingerprinting. In DNA fingerprinting, detection of the hybridized DNA fragment is possible by hybridized DNA fragment by the autoradiography. There is one process for the detection of the hybridized DNA known as the autoradiography. So correct answer of 180 is the by taking radioactive isotope. So autoradiography by the in DNA there is used 32 phosphorus radioisotopes. This is used here also. Now read question 181. So 181 mark the correct match. VNTR introns or exon? VNTR for introns. Variable number of tin and repeat for the intron only. Catalytic RNA and 23 as bacteria RNA as ribozymes. Wall found in eukaryotes. Lycopyron not found in eukaryotes, it's prokaryotes. So answer is variable ten and repeat. VRA for intron only. All of the following are part of an operon except an operator, structural gene, a promoter and enhancer. There is a operator in operator gene. There is operator present, structural gene, promoter, enhancer. There is no enhancer in the enhancer. And question is asking about operon. Operon concept is for prokaryotes. So the final proof for DNA is the genetic material come from the experiment of very easy question directly from NCRT or science change radioactive experiment take place take, take place by the Hersh and Chase 180 184. The association of histone H1 with a nucleosome indicates the association histone H1 protein with the nucleosome. What it indicate? The transcription is occurring, DNA replication is occurring, the DNA is condensed into a chromatin fiber. Condensation by the indicated by H1 histone protein only. DNA double helix is exposed, all other not answer. Answer is three answer. An association of H1 protein indicates the complete formation of the nucleosome, means condensation takes place. Which of the following RNAs should be most abundant in animal cells? rRNA. Most abundant RNA. rRNA only. Now section B. No, I took section B 186 question. During DNA replication, Okazaki fragments are used to elongate the leading strand towards replication, lagging strand. The lagging strand towards replication for away from replication form. The leading strands away from lagging strands away from replication. Answer is 4. 187. The lag operon is example of repressible operon, overlapping gene, arabinose gene, inducible operon. So lag operon is a type of inducible operon. So read. The two French scientists Jacob and Monod proposed the lag operon E. coli. The lag operon contains a promoter and operator regulator gene and three structural genes: ZYA coding for the enzyme beta galactosidase and permease and galactose transacetylase. So all these you can detail read here. So move ahead on question. The equivalent of structural gene is cistron structural gene which form DNA from DNA to RNA and RNA to protein formation take place by the DNA segment cistron DNA segment. Now read question 189. Which of the following rRNA act as a structural RNA as well as ribosome in bacteria? Important question. And directly learn ribozyme as well as catalytic structural gene as well as ribozyme. Structural gene as well as ribozyme. This only by 23S rRNA. 20, 23, 23. Yeah, this one 23S rRNA. Structural RNA as well as ribozyme enzyme also. So catalytic nature of the RNA we can understand by this example. In DNA dependent RNA polymerase catalyzes transcription of one strand of the DNA which is called as the which strand? Template strand only. Directly question transcription DNA dependent RNA polymerase DNA to RNA formation. Which enzyme? DNA dependent RNA polymerase. Which strand of the DNA take place? Template strand only. So now read question number 191. Which of the following required for as a inducer for the expression of the lycopron? Inducer. Lactose is inducer. So this is known as the lycopron only. This directly, it's very simple question. Which of the following is not required for the any of the technique of the DNA fingerprinting available like present? DNA hybridization, polymerase chain reaction, zinc finger analysis, restriction enzyme. Oh, what is zinc finger analysis? No take place here. Which of the following not required for the DNA fingerprinting? No zinc finger analysis process not used now. Zinc finger is a small protein structural motif they characterized by the coordination of the one or more zinc ion to order of stabilize their faults. So not used now. 193. Which of the following is the starter codon? Very easy. Everyone, I think everyone know this question. AUG is the starter codon. AUG is the starter codon. UGA, UGO, UGA. These are stop codon. So AUG, starter codon, and all other three. UGA, you can learn is UGA, UGA. UAA, UAG, UGA, they are stop codon. 194. Identify the correct genome. Genome is the largest from below. So largest to smallest. So here genome, genome, and here only genome. After genome, chromosome of nucleotide. Chromosome, okay, chromosome over, okay, here also chromosome. Then gene or nucleotide. Gene is the part of the DNA only, and nucleotide is the very monomeric unit of the DNA. So gene, nucleotide only fourth. Here nucleotide and gene sequence wrong. So read question 195. Satellite DNA is important because it's It's code for enzyme needed for DNA replication, codes for protein needed in cell cycle. I think not correct. So high degree of polymorphism in the population, also the same degree of polymorphism in an individual, which is heritable from a parent to children. This is the reason behind satellite DNA important. The codon causing chain termination are, chain termination means whatever we asking about stop codon. So UAA, UAG, UGA, UAA, UAG, UGA are answer of this question. Stop. They are stop codon. Removal of RNA polymerase third from nucleoplasm will affect the synthesis of. If there will be no RNA polymerase third, then which one of following synthesis will be affected? tRNA. tRNA formation by the RNA polymerase third. Synthesis of tRNA will affect by it. Removal of introns and joining of exon in a defined order during transcription is called as the which one this process? Splicing only, slicing, not slicing, splicing, looping, inducing, no, this one wrong. No, read question 199. Which one of the following is not a part of transcription unit in DNA? Which one of the following is not a part of transcription unit? You should now read question here. Transcription unit involved promoter, structural gene, terminator. So there will be no inducer. So answer here 3. Inducer is a chemical which will trigger the DNA to switch on gene in its presence. So transformation was discovered by directly question based on scientist. Griffith will discover transformation. Griffith experiment, first experiment only. So read carefully, do well, all the best.